type of samplers for seawater. So if the covered Vandon Niskin go flow, rosette is when you place the Niskins in a multiple bracket. Then you also have a Zobell sampler for sterile waters. Okay, the Zobell samplers one. Then I mentioned that it's sterile. Well, Zobell is actually the pioneer for marine microbiology. So the marine agar is the marine agar that everyone uses now. The commercial marine agar is actually uh, designed by concocted by Zobell. Okay, so initially the name was Marine Agar 2216E, but now they just call it Marine Agar. So he's the one that looked at the samples, seawater samples using a hemocytometer and and reported that the number of bacteria in the seawater is a lot higher than what is being cultured. Okay? So that was the beginning of the understanding that most of the bacteria that is in the environment remains unculturable. So we stayed here sterile. This is for sterile sampling. Okay? So it is based on a butterfly mechanism. Okay. So this is the cable. It's a it's like a a pair of butterfly wings. So what initially is there's a plastic bag in between the two wings. Okay, and then there's an inlet. Okay, so plastic bags can be sterilized, so they can be autoclaved, so they remain sterile. So of course, as with all samplers, there is a trigger mechanism here, and you can use a messenger to trigger it out. So when the messenger hits the trigger, the mechanism will open up the wings. So when it opens up, the plastic bag expands, then allowing the water to go into the plastic bag. Okay, so that's how essentially the Zobell sampler works. So if you look at it, it is a light design. Light. So it is quite difficult to control, especially in ocean if the ocean currents are strong. Okay. So if it keeps swaying around, when you release your messenger, it may not trigger the mechanism. Okay, because it keeps swaying around. So that's why even for marine microbiologists now, we seldom use this sampler. So if we don't use this sampler, what do we use? For marine microbiologists, what do we use? If we want to culture bacteria, we still use on this skin, we still use GoFlow. So are they sterile? Do they collect sterile samples? I mean, are they? Yeah, I mean they are not autoclavable, so there is no way you can collect sterile samples for microbiological work. But what the best practice is to clean properly, okay, and then whatever samples you obtain from here, whatever culture, uh, you have to use your own microbiological knowledge, your background to discern what are contaminants and what are actual marine bacteria. What are the actual marine bacteria in the seawater? Okay. If you're working in oceans far away from land, then if you consistently get Staphylococcus aureus or something that by nature shouldn't be in the marine waters, then you can safely discard it as a contaminant. Okay, because when you're far away from land, um, only the bacteria that can thrive well in high salt conditions will grow well on your marine agar plates. So that's how um, microbiologists will work around the problem of not being able to collect sterile samples. Collect samples using sterile containers. Any questions? Okay. So let's just say you've collected your sample. Then of course the first thing you gotta do is you have to either maintain. The first thing is actually the the best SOP. The best procedure is to immediately process the sample. If not, you have to maintain or preserve them. But this one depends on the kind of test that you want to carry out. If you want to culture bacteria from your seawater samples, then of course you cannot preserve it. You cannot put chemicals in it to kill bacteria. Usually we just keep it at 4 degrees Celsius. Okay, until we are able to culture them. If we are doing direct counting of bacteria, okay, for example, if you're doing flow cytometry, if you're doing epifluorescence microscopy, direct counts, then you can preserve them. Usually, you preserve them with aldehydes. Okay. The common ones are formaldehyde and butyraldehyde. Okay, so that's for bacteria. So for algae, for phytoplankton, they usually use Lugol's iodine, okay, because this will stain uh, the chloroplast, and you'll be able to see the phytoplankton clearer. But if you are working with chemistry, like if you do not, if you want to take preserve your samples for chemical analysis, then best is to freeze it. Okay. If you can't, if you don't have access to a freezer when you're, on a boat, then you can actually use mercury chloride. So this is, well, the step that we say is we poison it. We put mercury chloride into the samples. Okay. We do this to essentially stop the bacterial activity. So once the bacterial activity stops, the chemical concentrations, the chemical composition doesn't change. Because most of the change in the water chemistry is by biological activity. So by inhibiting biological activity, we can preserve the samples. Okay. The good thing about this is you can keep this at room temperature. So for long-term preservation, usually people use mercury chloride, especially when you don't have the facilities for a freezer. Okay. However, this one doesn't work so well for nitrate analysis, because it interferes with the one of the steps in nitrate analysis. But other than that, for ammonium, phosphorus, silicate, all the other common common nutrients that we measure in the sea, this is acceptable. Okay, an acceptable way to preserve your samples. Okay. We were talking about the importance of uh, close samplers, especially when you're doing trace metal work. So when you're doing trace metal work, also the wire that you use to attach your sampler should not use metal, should not be made of metal. Okay, you should instead use Kevlar coated wires because you do not want the metals from the wires to contaminate your sample. Okay, contamination can also be from the ship discharge or the leachate from the ship paint. Okay, so okay, now other than working with water samples, we also marine microbiologists, oceanographers are also interested in the sediments. Okay, why the sediments? Because the sediments contain what they call a historical record of the productivity of the waters above. Okay, because whatever production, primary production, secondary production, whatever activity, biological processes that are occurring in the water column, when the organisms die, they eventually sink. Okay, and when they sink, they create layers upon layers of historical record of what was happening in the waters above. Okay, so some that's why we it is also important to study the sediments, especially the surface sediments. So you have a gravity coral, you also have a grab coral. Okay, a grab coral is like this. Okay, you lower it down uh, with its jaw opened, And once it hits the sediment, it automatically, once it hits the sediment, it releases a latch. Then it will close up. Then when you lift it up, it will close and you will get a grab sample. So this is called a grab corer, or when wean grab. So what is required is you need it to be soft. The bottom have to be soft so that you can manage to grab the sample. Okay, but the disadvantages are when you do sampling like this, it significantly disturb the sediments here. Okay. You only collect the sediments, there is no overlying water. Okay, I'll show you how the gravity corer allows you to collect the overlying water also. Okay, so this is not designed to sample living organisms. Other than grab corer, you also have a box corer. Okay. So if you notice, there is no need for a messenger to trigger any mechanism. What triggers the mechanism is when the grab corer hits the sediment, it releases the latch that allows it to close the jaw of the corer. So here's the box corer. The two jaws are also here, you can see. But here you use a trigger mechanism. And this is attached to a wire. When you release the messenger, it will hit the trigger mechanism and release these two cables. And the jaws here are heavy, so once it's released, it will close. When it closes, it will capture a portion of the sediment here. But this doesn't disturb the surface sediments. So that's why the box corer still allows you to look at the surface sediments. Okay, so this is the makeshift grab corer. Then this is the trigger. So once this hits the sediment, it will release this trigger and will be able to close the jaws of the when wean coral. So this was we contracted the design to one of the hardware stores, one of the hardware stores in uh, uh, in Malaysia. However, the problem was it wasn't heavy enough. For grab coral to work, it has to be heavy so that it can penetrate deep into the sediments. Okay, that's why they added extra weight here. Okay. Uh, a gravity coral is essentially a metal tube. Okay, a long heavy metal tube. Okay. As I mentioned, it has to be heavy. All these corals are heavy because it uses gravity to penetrate into the sediment. If it's not heavy enough, it will not be able to penetrate deep into the sediment. So what we usually do is we place a PVC tube. Okay, we place a PVC tube inside the coral, and then we will close it with the, the mouth of the gravity corer. Okay. This one has a bit of a fan. So the design is uh, has soft teeth that allows the soil to enter but not come out. Okay, so this kind of like aluminum brushes that allows the soil to go in but prevents it from coming out. So from here, you release the coral. You release the coral deep into the sea. Once it penetrates, usually into the sediment, and then you slowly bring it back up to the surface. Once on the boat, on the ship, you remove the mouth. Quickly use a rubber band to cap the bottom of the PVC tubing. Okay. Then slowly you cap the bring the whole PVC tube out, and then you cap the top. So because it has penetrated into the sediment, you will have a layer of soil here. But the good thing about gravity coral is you will also have a layer of bottom water sediments, the overlying water in the tube. So science, those who are interested in the sediment processes, they are usually interested to study the interactions between the sediment and the overlying water. So some of the experimental design requires them to take several tubes like this. Okay. So what they do is they actually place an impeller to circulate the water, and then they slowly study the changes uh, in the water, changes in the surface sediments and activity. Okay. So a gravity core sample allows you to do that. Okay, so that's why it's an important way to sample the sediments. This also depends on the type of sediments that you have. If the bottom is hard, then you will not be able to sample much of the sediments. Okay, so the box coral, so the when wind grab coral is quite straightforward. So a box coral is like this. Okay, it has a trigger mechanism where you attach two uh, two jaws. Okay, so once the messenger triggers the mechanism, the cable will be released and the two jaws will closed. Okay, so that's why it will capture the sediments at the bottom. So when it captures the sediment at the bottom, it will close it up like that. Okay. Then once you bring it up, you can open up the cover here, and you will be able to see a nice undisturbed sediment surface. Undisturbed. Okay, this is important because the when wean, the grab coral uh, disturbs the surface. Okay. So this one will allow you to get the surface sediment undisturbed. So from there you can see the type of benthic organisms, their distribution on the surface of the sediment. Okay. When wind samplers do not allow you to do that. Okay. So other than water samplers and sediment samplers, you also use plankton nets. So is this a net with a weight? The important thing is the mesh, the size of the mesh. You can use a 20 micron mesh. You can use a 60 micron mesh. So the ones that I use in my lab is a 20 micron mesh. So what does this mean? That means what we do is we put the whole plankton net in the water to a specific depth, usually to the bottom, usually to the euphotic zone. So let's just say this is the euphotic zone. So this is the deep layer, deep waters. This is the euphotic zone. We'll just slowly put the net at this depth and then slowly lift it up. Okay. We know the diameter of the mouth here. Then we slowly lift it up. So when we lift it up slowly, we can estimate the amount of water that has actually passed through the net. So all the water that passes through here, okay, what is trapped here are those organisms that are more than 20 micron. So we are looking for organisms that are larger than 20 micron. So if you remember, 20 micron, larger than 20 micron, so we are looking at microplankton and above. Okay, microplankton fraction and above. 20 micron to 2 micron are nanoplankton. 2 micron to 0.2 micron is picoplankton. Then 0.2 micron to 0.02 micron, those are the femtoplankton fractions. So once we bring it up, usually they will use a hose to flush all the samples, all the microbes that are attached on the area here, so that it goes all into this container, weighted container at the bottom. Okay. Then once you have the samples, you can preserve the samples. Okay, so this is plankton net. However, from the design, of course, it looks flimsy. If the ocean currents are strong, there is no way you can pull it up straight. It'll be swaying left and right. Okay, so what do we do then? So usually in for those type of occasions, we don't use a plankton net. Instead, we use a makeshift concentrator. What we mean is just a normal bottle, but we make a hole here and place and stick a plankton net here. a mesh. Okay, so maybe a 20 micron mesh. So the water sample is collected as usual using Niskin, and then we pour a specific amount, maybe 2 liters of water here. So whatever is released from so the principle is the same. Anything that is smaller than 20 micron will be released. Okay. What is left are those that are larger than 20 micron, and they will be collected in the waters here. Okay. So that's how we can concentrate the samples, and when so that's how we can sample specific fractions of the microorganisms when plankton net is not desirable, cannot be used. Plankton net is used. Okay, so the bottom here is weighted. Okay, but sometimes it doesn't work because of the strong currents. Okay, so for those that are using 64 micron mesh, they are usually looking for zooplankton, copypods and beyond. Okay, so zooplankton net. For us, usually we go for 20 micron to look for microplankton. Okay. You also have a coarser mesh, larger ones, 250 microns. They collect the larger daphnias and the copypods. Okay. So this is the also a type of plankton net, but they call it the bongo nets. Okay, because of its two pairs, and they are used mainly for zooplankton and ichthyoplankton, so small fishes, larvae. You, you can see this at the mouth of the bongo net. This is a flow meter. So as the water goes through the bongo nets, this will turn and will tell you how much water has passed through the bongo nets. You need to know how much water has passed through the bongo net in order for your results to be quantitative. If not, your results will be purely qualitative. You will not be able to tell the readers how many zooplankton per mil or per liter of water. Okay, so this is unlike the plankton net where you lift it up from the bottom all the way to the surface. This one you drag, you tow. So the ship has to move and you'll be towing the bongo nets. So once the nets are up, you can use water from the ship. Usually this will be seawater because the ship will have a pump that pumps seawater directly into the supply. So you flush all the organisms that are trapped at the top so that it goes all the way to the bottom. So once you have the microzooplankton here or zooplankton here, then you can separate them into smaller bottles and preserve them. Okay, so as you can see, most biological oceanographers are happy. So she's now a lecturer at UCSI, University College Sedaya International. Okay, so this is the Van Veen grab. So this is actually the Vendon, not the Sniskin. This is a Vendon, but this is not the horizontal Vendon, but the normal uh, vertical Vendon sampler. Okay, it is not as good a design as a Niskin because sometimes it doesn't close as well. Okay, but the principle is always the same. Okay, there's always a rubber tubing in the middle to pull the two suction cups inside, and you always put a weight here. It has to be weighted. If not, your messenger will not work. Okay, it has to be weighted, and then once it closes, you can bring it up and you will collect the water. So this is the ship that we chartered long time ago, 2004, to traverse the Straits of Malacca for work in the to look at the water quality and water quality along the Straits of Malacca. So the whole cruise took about 7 days, I think. So that means we were on the ship for 7 days. So we have to sleep, bath, no, live on the ship for 7 days. Okay, so you're there, and when you're in the ship for 7 days, you can see the sunrise and the sunsets. So this is the Pulau Perak. Okay, this is the furthest. Uh, this is the island that marks the boundary between Indonesian waters and Malaysian waters, north of Straits of Malacca.